Great, super. Thanks very much. I'll try and stick to my 5 minutes. So my name's Amy. I'm a paediatric neurologist and a clinician scientist at Great Ormond Street Hospital in London and UCL in London. So my declarations. So in our project, what we've been doing is taking cells from patients with KCNT1 related epilepsy. So there are 3 patients, all in fact, who had the migrating focal seizures phenotype. We took skin cells and turned those into pluripotent stem cells. And we've also got 3 control lines that we've been working on. So what we've done is we've made a number of different model systems from the patient stem cells, and these include 2D excitatory neurons and recently inhibitory neurons, progenitors which have been differentiated into astrocytes, cortical spheroids and subpaleo spheroids, and then assembloids which contain both GABAergic and cortical excitatory neurons. So here in this initial work, what you can see is in the 2D neurons, we get good differentiation rates with more than 80% MAP2 cells at day 60 of cortical excitatory neurons. And we can see robust KCNT1 expression at day 60 of life, day 60 of differentiation as well. So this looks like a good model to assay KCNT1 in response to different drugs and therapies. What we can also see is that we do seem to get a bit of an astrocytosis, an increase in astrocyte production in the cultures, in the patient-derived cortical cultures, with an increase in S100 beta, which is a marker of astrocytes. And we've got some ongoing work in the lab looking at some cell autonomous astrocytic phenotypes which have emerged. So meaning that even in pure astrocyte cultures, we see some differences between the patient lines and the control lines. And we're now looking at exploring these more in co-cultures, and we'll have more information on these shortly. What we're also doing is we've made from control lines GABAergic neurons, as you can see here from control lines, and we're currently making some patient-derived GABAergic neurons again, so we can use these as a way to better investigate both disease mechanisms, but also look to use these as a platform for novel therapy development. And you'll be hearing from the CARDA group later, which is one of our collaborating groups that we're working on novel therapies with. I should say also that we've shown that we've got a good representation of the correct subtypes of the GABAergic neurons, including PV and other types of GABAergic neurons. So I mentioned that we're making 3D models as well. So we've used the Pascal protocol with these 3 patient lines to generate both cortical and subpaleo organoids. As you know, there's a number of different 3D kind of organoids that can be differentiated from stem cell lines. So we are making assembloids, which we make by making the 2 different types of organoids separately and then fusing those in a tube. And then what you get is you get migration of the inhibitory interneurons from the subpaleo organoids into the cortical organoids. So in some early work, we've seen that we get expression of mature neuronal and forebrain markers, which increase over time. So you can see here in the control lines on the left and the KCNT1 lines on the right, the expression of MAP2 and NUN goes up over time, and we get good expression of FOXG1, which is a forebrain marker. What we're starting to see in terms of staining and other phenotypes using QRT-PCR is that there seems to be an immaturity phenotype in the cortical spheroids from the KCNT1 lines, with persistence of progenitor markers such as SOX2 that we see in our patient organoids, and this is ongoing work in the assembloids. We've also been looking at functional assays in the 3D model. So we tried to do some single cell electrophysiology. That's technically quite challenging in the assembloids, which are quite heterogeneous in terms of what cell types are there. So we've gone for a local field potential approach at the moment, where we apply essentially an electrode to the center of the assembloid, and we then measure local field potentials both before and after the application of KCL, which is obviously relevant for the KCNT1 channels. And what we see in initial work is that for 2 of the genotypes that we're working on, we see a shift in the total band power compared to the control lines. And we see essentially what looks to be an emerging excitability, kind of pro-excitability phenotype in the KCNT1 assembloids, which have been in keeping with what we know from previous work. So at the moment, we are making CRISPR lines and looking to validate this phenotype using the CRISPR-corrected patient lines. That's ongoing work. We also plan to do MEA analysis in both the 2D and 3D models, and we have ongoing transcriptomic work going on to complement our novel therapy development. So one of the things I'm interested in as a clinician is that when I see patients with migrating focal seizures from different genotypes, be it SCN2A or KCC2, which is another gene I work on, they can have a different genetic cause, but exactly the same kind of epilepsy. So we wanted to see if there were common druggable pathways, which we could look at using bulk RNA sequencing between the two pathways. And we found some evidence of upregulation of oxidative phosphorylation and particularly of mitochondrial dysfunction, which we're investigating with further functional assays. And unsurprisingly, effects on GABAergic synapse formation, which have been downregulated, which are common between these two different genetic causes of the same epilepsy. So essentially, we have ongoing work in terms of phenotypes in both our 2D and 3D models. But I think what I've shown you is that they have many advantages in that we can assess lots of different patient variants in the kind of patient background. We can edit the patient lines with CRISPR to correct the mutation, but we also can knock in patient mutations to in-house lines. And what we're hoping through our transcriptomic work is that we can look at the impact of these variants in both the patient and the CRISPR lines on early neurodevelopmental genetic pathways. And then also the ability to, with our collaborators, look at the effects of rescue with different therapeutic approaches on the human transcriptome as opposed to animal models, which obviously these are a complement to. But there are some challenges, including largely cost and time, but also that this is quite a noisy system with quite a lot of heterogeneity between organoids, hence the need for the CRISPR controls is really important. So I'm going to finish there. Thank you very much.